don't like honey. It's always good for speakers. Sensitive. Could do with some caffeine.
I thought I could probably get some breakfast.
It's fine. Actually, if you could get some water.
I need some bottles of some sort. Do you reckon there's more bottles in the BDS stand? I'll go ask and grab.
Downstairs, one part on was water, wasn't it? It was just coffee.
I've got a tangerine.
It's too fiddly.
Yo, nice jacket.
You keeping people's time.
Don't know, I'm hoping the chair turns up and then I can sit down.
I'm sure they will do. They've got 10 minutes.
Exactly.
Yeah, you don't want to leave this to be self-policing. It'll go horribly wrong.
Yeah, and then if I found more.
Thanks for it.
Yep.
I'll
Thank you.
say
To
thank you.
be honest, they're all up ready for the
Okay, I'll let you.
Yeah, it's fine.
Well,
No. It's actually some people have carried on in science.
You just escape. They're just
So that I should do or
all done.
all right.
So they can just pull up their own if you want, or I'm happy to run between and
Okay.
pull them up, whichever you think's
Yeah,
easiest.
hopefully it will be.
Yeah, that's fine. And then also there's the clicker. Do you want me to go through with all the talkers how to use it? So that's a laser. That's back, that's forward. Don't use that 'cause
Yeah.
that brings up that. And that black screens, if they panic, just press it again and it will
All right.
reappear.
Fair enough. So it's back on. And you don't want me to press that one? Or?
You can. It's just back if you go too
All
far
right.
forward. But
So it's normally that, and the
yeah.
laser is. The
no,
one.
laser's that one.
All right. Yeah. Thanks. Oh, yeah.
And that
will.
one's blank screen. That
Yeah.
cuts the
Then
screen. Don't do
fair
that.
button. Yeah.
exactly.
Forbidden. Yeah. Okay. And that's the one we draw.
Yeah.
That's a shame. I was really interested in that one.
Yeah.
No. Anyway. Yeah.
Okay,
You see?
perfect.
No. Thanks.
I'll just be in the corner.
Yeah.
Should I ask the speakers or something if they want to be tweeted or non-tweeted? All right, so I don't. All right, yeah, you see? Yeah, we'll wait for them. Yeah. Anyway, that was something crazy. Yeah.
Am I leaving?
I just was briefed. This thing. Wait a minute. That's the laser, and that's the. That's easy.
So, are you Santiago?
Yep.
Did I pronounce that right? I'm Phil Wookie.
Nice to meet you. You are the first speaker.
I'm the first speaker.
Yeah.
I'm trying to keep this time, well, I will keep this time.
Yeah.
Because you can cut me off mid-sentence if you want.
I'm very relaxed in that sleep again. If I wake up, I start moving you.
If I wake up, that was a confident voice. Thanks. It highest height before is
standing up, sorry. No, sorry, that was.
There was no all the pointers though.
Yeah, there is one point.
Is that entitled?
Here. I will handle that. Well, actually, you take this.
So what's this? So it's
Yes.
backwards.
No, that one, no. The laser is that one. And then I guess you just put it again there.
Oh, let's just switch it off completely. Let's just forward the laser.
I will try to remember that. I'm afraid that's going to
Is the
happen.
laser not working?
No, but
It's a bit fine, isn't it?
Okay. And then that carbon, don't touch that one because I'm told you are going to arrow up
Towards
forward
backwards.
blank screen. If you do it, don't panic, just press it again.
So
If
put
it's
that
going
one.
horribly wrong, just blank screen it. Yeah, I think so. Do you want me to, I've got time on Wednesday.
Maybe worth just checking.
Yeah, last session.
All right. Yeah. Wait, wait, is it that? I call it traffic.
So where are you based?
Now in Bernard. I used to work in where we have
Well, everything in Spain.
Yeah, correct.
High mountains.
Not in Switzerland.
It's a bit more restricted, but there's
Yeah, it's
quite
very
big mountains.
beautiful,
I was in Switzerland actually in August, talking to Montuosa.
Oh, wow.
So we started walking, so I took the train from Zurich across to Bissau,
Yeah.
and then the post bus to Sassgrund, and started walking from there and ended in Sassgrund 9 days later. So
That's almost hiking
goes
one
on
month
like
that
that.
means yeah.
It was about 190 kilometres, something like that. So it's not 9 days, yeah. So it's alright, it's about 20 kilometres a day, but it's quite a lot up and down. Yeah, so there's punishing stretches, and also this is a very exposed stretches.
Yeah.
Just like
Yeah, that's one of the things I do is on hike and then there is a super thin path and
super thin path, nothing on.
that I want. And the level of stress of that thing is that
You just have to, I'm just going to, there's nothing, just in that way.
yeah, but if you are hiking, to me it's like so speed run.
Yeah, no, sometimes, yeah, you can see that. On the worst bits, there's sometimes some cables, just
Yeah.
sort of problems.
Right?
Maybe not. If there's no point to this, yes.
Yeah, well, just run out for that.
We can
And
just.
if not, you can always use the little mouse.
Yeah.
And what I'm gonna do is to load your talk. Which one is yours?
Santiago. So how long have you been in Bern for?
3 years.
3 years. Settled?
No permanent.
Back to Spain.
I'm
Quite big cultural differences in Switzerland.
no, I love it there, but Spanish has a very deep root, so I always want to go. But I'm also applying for things in
There's not a lot of
Switzerland.
jobs in Spain at the moment.
Not a whole lot, no, and the corruption level is pretty massive anyway, so 80% of the jobs are done.
It's a bit of a lost generation, isn't it?
Hopefully not. You know how to carry? Okay, you are very. All right. Yeah, if not, I can show with the map. By the way, Ruth.
I can manage that.
Ruth. No. How do you carry the presentation here? How do you carry it? Normally there is this.
No, I think it'll just be in.
Yeah.
Oh,
Yeah. The slideshow.
good. Where is it? Yeah. So, is it not just that one? It's a short.
All right. Yep.
Yeah.
Yeah.
Okay, welcome everyone to the session number 33. It's a very short name, so please pay attention. Ecosystem, ecology and functions. 2 points, biogeochemical cycles and plant soil interaction. So it's a bunch of very good talks. Unfortunately, one of them is withdrawn. Admor Mureva is not going to speak at 3:30. So we'll just have a gap of 15 minutes so we don't mess up with the organization of the rest of the conference. And the first speaker is Philip Walkie, who is going to speak about the effects of shrub encroachment on soil carbon in up north. So thanks, Philip.
Thank you very much, Santiago, and thanks to everyone for coming. It's always a tough one straight after lunch. So my talk is entitled Could Shrubification? Okay, it's now in the dictionary. And forest expansion threatens soil carbon stocks in the Arctic, and there's quite a big author list. And you'll note actually the first author here is Tom Parker, who's sat over there. But I'm doing the presentation because I'm based in the UK at the moment, and he did one yesterday, so that's pretty well the logic there. There's a pan-Arctic shrubification, an increase in cover and height of deciduous shrubs and subshrubs. This has been measured at many different plots. It's by no means universal, but it's a big-scale phenomenon. There's encroachment potentially as well onto tundra heaths by both deciduous and coniferous trees. This has been noted in the European Arctic, but is also occurring elsewhere. The big question is, will this productivity, this increase in productivity in the Arctic, mean that the Arctic becomes a stronger sink for carbon? That's the kind of logical standpoint that's taken by many of the kind of dynamic vegetation modelers. But as I hope I'll be able to point out in the course of the next 12 minutes or so, there's a lot more complicated stuff going on than this, and we need to think of the whole system. So, the Pan-Arctic greening, projects like greening of the Arctic from International Polar Year and subsequently, we know now from things like MODIS satellite imagery that there's a greening going on. And these are data from 1982 to 2011. This is a vegetation index trend. It's based on NDVI, so Normalized Difference Vegetation Index, which is ground truth by people like Howie Epstein in different locations around the Arctic. These are data from the Yamal Peninsula there. So there's greening going on, but again, this isn't universal. There's some browning in some areas, as you can see, productivity declining potentially. But this is important, and this matches and corresponds with experimental studies like the International Tundra Experiment, which has produced a lot of quite big-scale syntheses over the years of what happens when you warm tundra plots, both in the Arctic and the Alpine, to productivity and other measures of plant success. Phenological responses, reproductive output, things like this. Other papers that I could have included there are people like Isla Myers-Smith's work on shrubification as well. And this is what it looks like in some of the low Arctic areas. This particular shot is taken in Northwest Territories in Canada, quite close to, well, between Inuvik and Tuktoyaktuk. This is green alder, okay, in the foreground, and there's dwarf birch in the middle distance there that's turning autumn colours. And this is a process of greening which has been recorded in many different sites around the north now. However, vegetation change, of course, is the tip of the iceberg, and this is certainly true in tundra environments where most of the actual biomass is below ground. Okay, 90%, 85%, this kind of thing. So we really do need to understand plant-soil interactions, soil organic matter dynamics, and greenhouse gas fluxes there. So we really need to get beyond just looking above ground. If you don't believe me, here's some of the data from the Northern Circumpolar Soil Carbon Database. Okay, there's a paper published in 2013 by Gustav Hugelius. And I want you just to notice the units here. So this is in the permafrost region, so this strays into the boreal as well, rather than just Arctic tundra. But in the darkest of the browns and so on, the reddy brown there, we're talking about up to about 100 kilograms per metre squared. Okay, so effectively me's worth. Of course I'm not 100 kilograms, how could I? Effectively me's worth of carbon, okay, per square metre. Okay, so there's a lot of carbon in there, worth the equivalent of about 250 years worth of fossil fuel carbon combustion now. So big deal. So if we now consider the greening, so what we measure by satellites against the actual stocks of carbon, I think you can see just if you compare the units on left and right, we're dealing with grams per metre squared on the left, and that's per year, so that's change in phytomass up to the areas greening most, about 10 grams per metre squared per year. But set that against the context of how much is actually below ground, and this is in permafrost regions, but this extends beyond that as well in cool and often wet and waterlogged situations. So we need to be aware of that. So if we think about climate, how it's exerting its impacts on ecosystems, there's an above and below ground component to this, obviously. So there's an interaction between climate and plants in terms of trace gas fluxes, physiognomy, reflectance, surface roughness, that kind of thing. There's an interaction with soils as well through things like trace gas fluxes. That's sort of well established. I would argue, and someone can burn me at the stake later for heresy, but I would argue that if we think about climate change effects on plants, we actually know quite a lot about this, and we can model fairly effectively in the circumpolar north the relationship between things like foliar nitrogen and leaf area index, and get a fairly good handle on what's happening in terms of gas flux into the system. Okay, so I've put a tick-ish there. We kind of know that reasonably well. Soils are much harder in terms of how they respond to warming and what that means in terms of nutrient release and trace gas fluxes. There's issues there like acclimation to warming and all sorts of things that have occupied quite a few people, including myself, in their careers. But it's this component of the interactions that's a really, really kind of key aspect that we need to try and understand. Okay, so the direct responses of soils to changes in thermal and hydrological regimes in the Arctic, they're challenging enough to predict on their own. But it's these plant-soil interactions in the rhizosphere that may exert a critical control over ecosystem carbon stocks as well. This is particularly the case in an environment where we've got huge amounts of carbon in soils, and we've potentially got shifts in vegetation occurring or underway at the moment. So my interest in this all really started when I was based back in Uppsala in Sweden back in the 90s, and I had a PhD student, Sofie Högerstein, there on the left, who was doing her PhD as part of a broader EU project on tree line dynamics, and we were responsible for the soils bit. And we kept noticing, and we hadn't really had a clue beforehand this was the case, but we noticed from her plots that every time we went into forests, we had very, very little soil carbon. Every time we went out of forests and onto heaths, we had a lot more. Okay, so this was the deal there. It's just a couple of sort of like, you know, quick profiles there, and the scale there is about 10 centimetres. Okay, so this is the forest. We're talking about centimetre, centimetre and a half in the organic layers. Okay, there's organic material below that too. This theme was picked up, so Sofie did a lot of really neat work on that, and then in a subsequent project as part of International Polar Year, Ian Hartley, who's now based in Exeter University, picked up on these themes, and we really got to town on unravelling what was going on. So in Sofie's project, we noticed through a whole transect across Scandinavia, from Dovrefjell in south-central Norway up to Jotkajärvi in North Norway and Finnmark, via Abisko and Vattejärvi in Sweden, that we always found we had more carbon, okay, in heath compared to forest plots. We also, through CP mass 13C NMR analysis, one of the first applications of this actually in this environment, we found carbon's more labile as well in the heaths. Okay, so that's an important thing. So Ian, in his project as part of International Polar Year, he did some really neat work on trying to understand what was happening in terms of soil organic matter turnover in heaths and forests. And this is the result of that. We deduced that we actually had a loss of carbon, potentially, at whole ecosystem level if you switched from heath to forest. So in other words, if you go from the left to the right there, does that actually represent a loss of carbon? Well, it doesn't look like it if we look at above ground processes. But if we look at the actual carbon stocks on the top, the bottom's nitrogen, the left-hand block is tundra heath, the right's birch forest, and you see the zero demarks a ground surface. So we've got above and below ground. You can see in the forest, sure, we've got more carbon above ground, but we've got substantially less below ground. And if you add them all up, the heath is actually more carbon rich. Okay. So we did some nifty stuff with our links through to the NERC radiocarbon facility at East Kilbride, and the deputy director of that, Mark Garnett. And we were doing work there on radiocarbon analysis of respired carbon dioxide in relation to the bomb peaks, so hence the mushroom cloud there of 14C injected into the atmosphere in the late 50s and early 60s. And with that, we were able to deduce that we were getting a faster turnover of carbon in the forest, much faster compared to the heath. Okay, so this is what it looks like in this kind of area. We've got ericaceous heath there, you've got some forest patches and so on. And this is where Tom's PhD comes into the equation. So this is a paper that came out a couple of years back now. And this is about carbon turnover in shrub and tree plots compared to heaths as well. And as the title suggests, we've got very rapid carbon turnover in this context. And so from Tom's PhD thesis, it's really a question of comparing trees and shrubs here, so deciduous in this case, with ericaceous heath, with empetrum on there, so crowberry. On the left there, we're dealing with ectomycorrhizal situations. On the right, we're dealing with less of that, predominantly ericoid mycorrhizas. So inside the forest, we've potentially got deeper snow. We've got more benign conditions in the winter under the snow. We've got better quality litter, so everything's turning over faster. So shrub and forest dominated systems store less soil carbon than adjacent heaths. Carbon, we hypothesize, is cycled at higher rates in more productive systems. Rhizosphere priming is something we think is very important. That's a stimulation of soil organic matter decomposition by live roots and associated rhizosphere organisms. Okay, and this is enhanced by ectomycorrhizal hyphal growth, okay, which is very much explorative through soil organic matter compared to ericaceous, which is much more closely associated with roots. So this is what we're talking about. So there's Lorna Street, so for scale, and there's a sporocarp there of Boletus, and we're talking about these kind of mycorrhizas with hyphal networks stretching a long way away from the host plant. So from Tom's work, we find here, this is a transect from heath on the left into forest of organic matter stocks, okay? The brown is the organic horizons, the rest is the mineral. So very much more carbon in the heath. This compares data from Abisko, which is one of the driest places in Sweden, just in case you thought it might be a hydrological thing. If we do this a few kilometres to the west at Vassijaure, this is one of the wetter places in Sweden, we see exactly the same thing. We also note through respiration measurements that the forest soils respire much more readily, and also the shrub soils. So the darker the greens there, the more foresty it is, and the browns here are the heath, okay, at the bottom. And this is corrected, and it's expressed per kilogram of soil organic matter as well. So we've got very much more rapid breakdown going on. Okay, perfect. And so if we now go and measure hyphal ingrowth, this is what's the data that we've got here, we can see something similar at the forest edge. We've got most hyphal ingrowth, okay? It doesn't differ significantly between forest and heath, interestingly, but we suspect very strongly that something is going on. So we think as well, this is what we're looking forward to by 2050 in terms of climate envelopes around the circumpolar north, a shift towards more ectomycorrhizal species. And just bringing this to the more recent work we've been doing in the Northwest Territories in Canada, we've been working in a place called Trail Valley Creek. And this has involved quite a big team, but the person leading this is Lorna Street, so in the middle there. We've been doing 13C pulse chase work, natural abundance work with 14C, and conventional gas exchange measurements. And we find again that highly productive shrub communities also recycle carbon very rapidly. They may not represent an increased carbon sink. So that's there's some of the data there that underpin that. I won't worry too much about that, but just as a quick sort of like overview, the left is productive shrub or forest, green's photosynthesis, RS is soil respiration, SOC is soil organic carbon, rapid turnover, not much soil carbon. So if we switch that onto a tundra heath and we colonise the heath, so we just move that over, okay, we're effectively putting more carbon in, but we're accessing more carbon stock below ground by the ectomycorrhizas, okay? And we're potentially pushing out, accelerating the release of carbon through priming to the atmosphere. So the whole system actually becoming a net source, okay, of carbon to the atmosphere, counterintuitively, okay? Even though productivity would go up, that's a little bit cheesy, the graphic there, thanks to PowerPoint. I'm not suggesting all the organic material is going to be respired. So thanks very much. The acknowledgements are to a whole host of people, starting back in 1995, so 21 years ago in the International Tundra Experiment, through several programs as well. And our partners currently are these in Canada for our current project, so another cast of thousands. Cheers and happy Christmas.
Thanks, Philip, for this interesting talk. We have time for one question over there.
Yeah, I'm just interested to hear your thoughts on upland heaths in the UK as potential analogues for those sorts of Arctic systems, and whether, in principle, the same processes could apply with tree invasions taking place in upland heaths in the UK.
Yeah, thanks, Alan. That's a fascinating question, and it's one that we actually have given some thought to. And we're kind of like hoping maybe that we can start some work with JHI folks and the Morco plots, for example, in Scotland on some of these things. It's absolutely an analogue, for sure, no doubt about that. One of the things that's attractive about working, certainly not in Canada, because that's a permafrost landscape, but the Abisko plots, there's no permafrost there and it's relatively freely drained. So the heaths that we're comparing are sort of mesic with the actual forest adjacent, which is mesic. So we can tease out effects of rhizosphere processes from hydrology. In Scotland, we've always got the interesting thing that perhaps as soon as you put birch onto Calluna moorland, then effectively you've got transpiration as an important control, potential drying as well. So it's potentially very, very important. In Scotland, you could say it's even more of an issue, potentially, because you've got that double effect, which might very well be important. In the context of Arbisco, there's actually, though I've played up the actual amount, the stocks of carbon in the heath, it's very low compared to many of our uplands in the UK.
Okay, thank you very much.
Thank you.
Next speaker, we still staying on the Arctic. Andrew Martin, you're going to speak about...
very similar topic. That was a really nice
introduction. Yeah, all right. There's some space in there. So the exact title is Evaluating Approaches to Infer the Mechanism Driving Plant Growth Responses Over the Long Term Using Wood Rings. An example of the role of soil nutrient availability on Arctic shrub growth. Very short and cozy, Andrew.
Sorry.
Okay, well, thanks.
That's it. Yeah. Yeah. Okay. Okay, hi everyone. Very similar topic, and I think a lot of my introductory material can probably be skimmed through now. I'm a PhD student at the Long-Term Ecology Lab, University of Oxford, with Kathy Willis, Mark Mashus, and Lizzie Jeffers. And I wanted to talk to you today about a new approach I've been developing, a mechanistic modelling approach using dendro-ecological data sets to look at long-term shrub growth, soil interactions, specifically soil nitrogen availability. So we know what Arctic greening is now for the people that have just been in the room, but it's the remotely sensed increase in greenness of pixels, which is largely explained through an increasing dominance of deciduous shrubs at the expenses of mosses, lichens, and evergreen plants. And we have evidence from satellite observations from the International Tundra Experiment plots and ethnographic study that support this interpretation of a pan-Arctic shrubification, which occurs through three major mechanisms, these being the northward advance of shrub species, their increasing height, and infilling of patches, for example, on a azonal landscape where you might have riparian corridors of salix species, for example, moving outwards into the landscape. So is this greening / shrubification driven by climate? Recent analysis from Piao et al. 2014 demonstrated that the partial correlation coefficient of the NDVI or greening signal and temperature has decreased progressively over the past 30 years from about 0.75 to 0.3, I think. And this kind of suggests a decoupling between the climatic drivers and the trends that we're seeing. And also very recently, the time-integrated NDVI suggests that we've seen in some areas, especially Eurasian regions, a reversal of the greening trend and browning. So the question I'm trying to answer is, what are the possible alternative explanations for the shrubification trends that we see with regard to other potential drivers? And so something controversial that I'm interested in is progressive nitrogen limitation. This is the idea that when you have increasing atmospheric carbon dioxide, a stimulation of woody biomass specifically occurs as shrub plants invest in structure, which can potentially lead to a transactional progressive nitrogen limitation as there's a decline in the quality and quantity of leaf litter nitrogen. And we suggest that this could occur in Arctic regions because there's a low input of nitrogen, a relatively low input of nitrogen through nitrogen deposition. And also we have some evidence to suggest from sediment core delta 15N isotope values that there's been a stagnation or even decline in Arctic catchment scale nitrogen availability over the past 100 to 150 years. Okay, so my core research objectives are to understand at a kind of fine scale how soil nitrogen availability has changed through time as well as in a kind of local space. To what extent the growth of tall shrubs has been constrained by any kind of effects of nitrogen limitation, and is there any evidence for any plant-soil positive-negative feedbacks between soil nitrogen availability and shrub biomass? And so the main methods I'm using are dendro-ecological approaches to reconstruct nitrogen, soil nitrogen availability and shrub growth through secondary growth, and using mechanistic modelling approaches to try and understand the mechanisms that might be occurring in these systems. And so some of the methodological challenges I've had during this process have been with the nitrogen reconstruction itself. It's a technique that hasn't really been used on a long term in the Arctic, and associated with that is a temporal resolution of data I can achieve, and then developing a mechanistic modelling approach for this problem and understanding process variability through time. So just to talk about the study design, I'm working in the Yamal Peninsula in Arctic Russia, and I've got 4 sites along a latitudinal gradient where we've collected 40 shrub individuals per site. So this is the wood stem disc. So poor shrubs have been kind of cut down and we've harvested them. And from these, I'm reconstructing the growth using ring widths and wood densities, and the stable nitrogen using delta 15N values. And this proxy has been validated, for example, by Kendra McLaughlin with multi-proxy approaches between sedimentary delta 15N and wood delta 15N. And just for today, I'm just going to talk about Varandi, which is don't know if I've got the one over on the left, which is where we would, the reason I've worked on this site first is because it has the largest growth rings. It's the least harsh of the environments, and that enables me to work with the developing the methodology of reconstructing the nitrogen. So briefly, I have, these are our growth chronologies, which were created for another study, Methodological 2012. And just to highlight the kind of nature of growth of these shrubs, this is the absolute growth rate with millimeters through time. And as you can see, there's a kind of period of growth suppression for a good 20 years on most individuals at the start of their life. And we think there's some kind of growth suppression due to shading or environmental competition, just to bear that in mind. So to create the nitrogen chronologies, we do this a delta 15N approach, and there are some specific challenges in this environment. Firstly, the rings are really hard to see because they're so small and very gnarly, shall we say. They don't grow geometrically. So what I actually do is I use microtome to make wood anatomical slides, and I also take the opportunity here to identify discontinuous features such as frost damage and herbivory damage in the shrubs. For the technique, you have to use the bulk wood for a sample, which, so normally in isotope analysis, like with delta 18O, you would identify a component where that element is fixed. Unfortunately for nitrogen, no one has achieved this yet, so we use the bulk wood, and this means we need a large sample size. So it's not possible on that small suppressed growth period to achieve annual resolution at this time. So I'm doing 3-year bins of nitrogen data. Seeing as how I'm looking at long-term trends anyway. And I'm also going to look at the spatial heterogeneity because these are point-based readings. So these are my first 5 nitrogen chronologies from this site. They're the orange lines. I don't really want to try and wiggle match explain them to you, but you can see that they're doing quite different things, as suggested by a very large negative signal-to-noise ratio. And the small box plot here is just to demonstrate that I've tested different methods of homogenizing the samples, the big samples, to make sure that there's no homogenization issues with the readings of delta 15N. So my modeling approach is, the main characteristics of this are I'm using a maximum likelihood model fitting and model selection approach. And I'm also adapting this to use a local likelihood method so that I can assess the variation in model parameters through time, and this enables me to infer if I've missed biological mechanisms from my models, and then either refine the models or try and explain them. To do this approach, I've developed a small library in F#, if anyone's familiar, which I've tried to include a very robust optimization routine because I found because our data is so noisy, there was a lot of local minima in the problem. I calculate confidence intervals using a bootstrapping technique. I have that local likelihood approach in there, and I also have constrained optimization there, so I can constrain parameters to be biologically meaningful. For example, using a logit, so they're between 0 and 1, or logging, so they're above 0. And also in development on this, but I don't have to show you, is a small time delay approximation. approach, which means that although I've got 3-year nitrogen data and annual growth data, I can then run my model fitting and model selection on an annual resolution to increase the power of so I'm not throwing away data effectively. So I'm using a nested model approach. So if you imagine I've just got if you imagine back to the stuff about progressive nitrogen limitation, I've got kind of 3 areas of hypotheses here. I've got growth that's nitrogen independent, nitrogen dependent growth, and nitrogen dependent growth is some kind of plant soil feedback. And I'm interested in making sure that the growth models I select as the basis for these models best explain the growth of these shrubs. So I'm varying that. I'm also varying the functional form of the nitrogen limitation. So in terms of the plant growth models, if you imagine you've got these basic kind of shapes of plant growth, you could argue that given the shrub data I showed you earlier, the generalized logistic or Richards curve would be best because it allows for a flexible lower asymptote, so it enables you to have that modeled low growth period before a release. And I do see that in the data. So this graph here is AICC values, which are the IKE's information criterion values. The lower the better. So you can see that the logistic is really bad fit for these data. So that suggests which growth models to take forward. This panel on the right here, you can just see a demonstration of those growth models fitted to an individual shrub and the values. So this, the Gompertz, for example, is predicting a maximum radius of a shrub individual of 48 millimeters there. So moving on to my hypotheses, these are developed as coupled differential equation models. So as you can see here, the top equation is the change in mass through time, and the bottom equation is the change in nitrogen through time. And I've modeled into here the fractionation effects moving from the proxy to the real nitrogen value. And nitrogen limitation is included through a 0 to 1 modifier, which is A in this problem, which operates on a Gompertz gradient. And just to show you, so I've just run that hypothesis model for all 5 of those shrub nitrogen chronologies, and you can see we get these kind of real biological variables out, parameters out of the model. Although these I ran with a logistic growth curve, so as you can see here on the right, the fit's actually quite poor. So, but I just wanted to show you the kind of approaches that I'm using. And so going forward, I just need to refine these model systems and then compute all of the IKE information criterion for annual time series, do some analysis of spatial environmental covariates, and do some analysis of parameter variation. Thank you. And if you're interested in pollen, we have a website, globalpollenproject.org, that I made as well, which is a crowdsourced pollen identification database. Thank you.
Very interesting talk. Do we have any questions?
You showed that nitrogen plays a key role in growth. I just wonder then how at the beginning you came to arrive at assuming CO2 has an effect. I don't know no empirical data that support that.
Well, I'm just planning to test a hypothesis, but I mean, that's what base experiments are for.
But that hypothesis had been falsified for the Arctic and for the Alpine and for any natural understood system, except for some dry clusters. I'm just saying it because it's such a popular assumption and it's simply wrong.
Okay. Yeah, I don't really have an answer to that.
Yeah.
What are the rates of nitrogen deposition at your study sites? You said they're not very big.
So were you at the functional ecology talk for lunch? So the maps that were shown there. So it's I'm not sure of the exact amounts. I just know so for the 2 forms of nitrogen, one is extremely low and the other is very low. But I couldn't tell you exactly. But that would be something I'd be interested in incorporating into the models.
Yeah, just given that the Arctic is such a limited system nutrient-wise, even a small addition could have quite a big impact on the community.
Yeah.
Thanks, Andrew. Well, just a double check. I thought it would be withdrawn, but Admor Mureva is not in the room, right? Okay, yeah, I imagine so. So we have 15 minutes for free. If someone has more questions for Philip or Andrew, or just leave the room. Just remember to come back for next division, which will be the next talk. Thanks, guys.
Cheers, yeah. It's a lot of
Depending on that, I'm pretty sure we have to use that.
I think it's 2009 or 2010. And there we show
Don't get nervous and push the center. That's not the laser. That one is the laser. If you push that one, the presentation goes away. We need to restart it. All right? That one and that one. That layer. What? I mean, you panic just push it again. If you just mess up with a button.
That's actually what I'm saying as well. That's actually what I'm saying. I know. That's something my career. But now I'm not in academia anymore, so I'm not going to say whatever I want. Yeah, I did. Yeah,
I'm sure I can add expenses. Hi, welcome back to the session on ecosystem ecology and function. Now we restart the session with Noemi Pichon, which is going to give a great talk about a project that I never heard of. And she's casually from the University of Bern. And well, she will present the direct and indirect effects of nitrogen enrichment on grassland ecosystem functioning. So thanks, Noemi.
Thank you, Santi. So I'm very pleased to present to you today the first results of our PanDiv experiment, on which we are trying to disentangle the direct and indirect effects of nitrogen enrichment on the grassland ecosystem functioning. So, as you know, nitrogen enrichment is part of global changes, one of the major drivers of global changes. And the nitrogen levels have been increased in the ecosystems through human activities like organic and inorganic fertilization due to agriculture, of fossil fuel emissions. And nitrogen enrichment has a huge impact on ecosystem functions It increases the biomass output, it changes the soil chemical properties, and it also has an effect on biotic communities. Here on this graph, you can see the different effects of land use intensity, of which nitrogen enrichment is a part of, on the ecosystem multifunctionality according to different sets of function on an ecosystem. So nitrogen enrichment has effects on biotic community. It reduces the species diversity here on the left. And it also has an effect on the functional composition of the ecosystem. So when we fertilize the ecosystem, we are seeing a shift of the community towards a more resource acquisitive strategy of the plants. So what we call here fast-growing species in comparison with a more resource conservation strategy, so slow-growing species. And we also can see an intraspecific shift of the plant traits. This is what I will talk about later. This shift in biotic community have also great importance on ecosystem function, but the impact of all of this direct and indirect function is not really well known. So what we're trying to understand here with our experiment is to really see the relative importance of direct effect of nitrogen enrichment on ecosystem function in comparison with indirect effects through a change in the biotic community. In our experiment, we're also adding a higher trophic level with fungal foliar pathogens, but I'm not going to talk about this today. This is the framework of our project, our whole project, and I will explain you these relationships here with taking as an example the biomass output as the main measure of our ecosystem function this year. So the first arrow here is the direct effects of nitrogen. So when you have more nitrogen, it leads to an increase in the biomass production. The first indirect effect is the biodiversity ecosystem functioning hypothesis. When, so nitrogen enrichment leads to a decline in plant diversity, which reduces the biomass output. And this indirect effect is the resource economic hypothesis, which states that nitrogen enrichment leads to a shift in the plant community towards species with more exploitative metabolic strategies, of more so fast-growing species. And these species tend to have a higher biomass. So this indirect effect increases the biomass output. So what we're trying to understand, our question is here is to see the different shape of this free arrow here. So to answer this, to answer this question, we were setting up an experiment last year near the city of Bern in Switzerland. We have a pool of 20 species, 20 common species, which were separated in fast and slow growing according to plant functional traits. And we are growing the species in monocultures in 4, 8, and 20 species mixtures. And from in these mixtures, we have only fast growing, only slow growing, or a mix of fast and slow growing. So in total, we have 84 different combinations of species that we're growing 4 times, one without any treatment, one with nitrogen, one with fungicide, and one with nitrogen and fungicide. So this whole field, the whole plots are weeded 3 times a year to ensure the same composition through time. And the field is mowed 2 times a year to simulate extensively managed grassland. So we apply 2 times per year, we apply urea, and we apply fungicide 4 times a year. So now I'll present you my results, first the biomass, and then the intraspecific variation due to our 4 manipulations here. So the biomass data were collected this summer. They were collected in all the plots across the field. And these data are corrected for the species percentage cover. So these are data for biomass without the weeds in the plots. We can clearly see an effect of species diversity, so monocultures for 8 and 20 species, an increase in biomass. And there's also an effect of nitrogen on the biomass. I was trying to see if I could see an effect of the nitrogen on the realized diversity, so taking into account the weeds with our target plants, but I couldn't see a significant effect yet. And what is a bit disappointing as well is that we are not seeing a clear effect of the functional composition on the biomass output or an effect of the community weighted mean plant traits that I will explain just now. Before I show you the results for the interspecific variation, I just want to show some expectations I have about my data. So I was measuring the specific leaf area across the subset of the field, and so I took 5 samples on all the species on the subset of the plots of the field. So here we have functional composition. Fast-growing species tend to have high SLA values, and slow-growing species tend to have low SLA values. When we add nitrogen, we expect higher SLA values, and we could also maybe observe a higher intraspecific variation for fast-growing than for slow-growing species. And when we add with this the species diversity, so monocultures 4 and 8 species, we can see also different patterns according to these factors. So now this is what I observed. These are the results for the specific leaf area values without taking into account the community weighted, the species percentage cover. So this is just the mean value of the specific leaf area across the field. There is an effect of nitrogen. The nitrogen increases the SLA value. And there's a clear effect of the functional composition on the plot. So fast-growing species have higher values than slow-growing ones. And this is coupled with an effect of the diversity level. Here, an increase of the SLA values with the diversity level. And here we see actually a higher, we could maybe see what seems to be a higher intraspecific variation here for fast-growing species. When we take now in account the community-weighted mean specifically leaf area, so when we take into account the percentage cover, this is what we observe here. So we still have an effect of nitrogen and of functional composition, but with diversity we see a shift of the plot community-weighted mean specific leaf area towards for higher diversity levels. This could be explained by the fact that one or a group of species with especially low SLA values have a higher percentage cover than others, are establishing better than others. And when we take a closer look at this, composition, the 8 species slow-growing plots, this is actually what we observed, that one species or group of species, here for instance Achillea millefolium in red, has a really low SLA value and very high percentage cover compared to species like, for instance, Anthoxanthum moderatum that have higher SLA value but really low percentage cover. This could be explained by the fact that these species that are here have been really well establishing at the beginning of the experiment, and since this is the very beginning of our whole experiment, the soil was pretty dry this summer, and having a low SLA value could have favored the establishment of this species compared to this one, maybe. But we can maybe discuss about this a bit later. So to conclude about the stock, we've seen an effect of species diversity and of nitrogen on the biomass output, but we can see a clear effect of functional composition or of the community-weighted mean specific leaf area on the biomass output yet. There is no clear effect here of nitrogen on realized diversity yet, but these, again, are maybe patterns that we'll see in the future. And finally, there is an effect of nitrogen, species diversity, and functional composition on the intraspecific variation of the species on the specific leaf area. And we observe different patterns when we look at the SLA alone or the community-weighted mean SLA. So, yeah, even if it's only the first set of results for this year, this shows really the importance of these kinds of experiments to really understand, fully understand what are the relative impacts of direct effects and indirect effect of nitrogen enrichment on the ecosystem function. Thank you very much.
We have plenty of time for questions, so yeah.
Do you have any preliminary results of the effects of the fungicide yet?
Yes, the fungicide increases the biomass output, and yeah.
How did you apply the fungicide?
We're spraying the fungicide on the leaves of the plants and the plots without with avoiding the soil.
Okay, let's talk more detail later.
Okay, but this is not my topic in the but yeah.
Any more questions? Yeah.
In one of your results, you showed biomass production, and you said that you eliminated the effects of weeds, which you found in your plots. Like, can you elaborate a bit more? Like, did you find many weeds, and was that different between the different species compositions, so with different diversities?
Yes. So we're measuring the weeds percentage cover in all of our plots. We have around 100 different weed species across the field, and we have quite a large amount of weeds in the monocultures. And this, so the percentage cover of the weeds decreases with increasing the diversity level. So in 20 species cultures, we have really low percentage cover of weeds. And we have weeds that we're establishing at the very beginning of the experiment that won't be a problem anymore because they're not used to high competition. And yeah, so we try to we will see in the next years we have one part of the plot that is left unweeded. Here you have a plot, for instance, and this part above is not weeded. And here we will measure the weed community, and we will see what is inside and how it changes with the plants that are in the plots. Yeah.
Okay. Thanks, Noemi.
Thank you.
You are free in the rest of the conference. So next we keep going to grasslands and nitrogen with Aneta Trajanov, which is Trajanov, sorry, which is going to present the predictive mineralizable nitrogen in grassland soil. So thanks, Aneta.
Thank you. Okay, so what I'm going to talk about now is a bit different from what you've heard so far, because I'm going to present a different approach towards modeling and predicting mineralizable nitrogen in grassland soils. So why do we need to predict mineralized soil nitrogen? You probably know that mineralized soil nitrogen is an important source of nitrogens for grassland production. Some soils can supply large quantities of plant-available nitrogen through mineralization of soil organic matter. And grass grown on such soils require less fertilizer, nitrogen fertilizer applications per year. So improving the nitrogen fertilizer use efficiency in grassland soils is important for agronomic and environmental sustainability. And therefore, the supply of nitrogen from soil reserves, which is obtained from the mineralization of nitrogen in the soil, is very important when planning future nitrogen fertilizer applications in the field. So that's why we want to predict the mineralized nitrogen in grassland soils. The data set that we used in our study comes from Chagask from Ireland. It consists of soil samples from all around Ireland. It consists of 93 soil samples of permanent grassland sites. The soil samples were described by a broad range of soil types, diagnostics, and physiochemical properties. These samples were taken from March 2012 until October 2014 and were taken for all surface horizons for the depths of 0 to 25 centimeters. So each soil sample was described by different chemical, physical, and diagnostic properties. So here you can see, some of the chemical and physical properties. There were many more. So for example, we had percentage of total nitrogen, percentage of organic carbon, pH, relative proportions of sand, silt, and clay particles in the soil, etc. The diagnostic properties were described with the following variables: wetness, organic matter, depth, argilicity, spodicity, and calcareous. So what we wanted to see in our study, we wanted to see how informative different types of soil properties are for predicting the mineralizable N in the soil. So we actually divided our original data set into 3 sets of data, where we first used only chemical and physical properties of the data of the soil. Then we used only the diagnostic properties, and then we combined all chemical, physical, and diagnostic properties together. And then we also created 9 scenarios which presented different combinations of input parameters, and these 9 scenarios were created for the chemical and physical properties only, and for the combination of all properties. So we wanted to predict the quantity of mineralized N in the soil, and to do that we used data mining methods for predictive modeling, and more specifically model and regression trees. So a brief introduction to data mining. It is part of the process of knowledge discovery from databases. It is actually extracting knowledge from databases in an automatic way. So the process of data mining follows these steps. First, we need to have a case study, a problem that we want to solve, and normally data. Then we need to decide what kind of patterns do we want to find in this data, in what kind of format do we want the knowledge from the data to be presented. Then we usually need to pre-process the data set in order to present it in the format needed for data mining, for using the data mining algorithms. Then we apply the data mining algorithms and we obtain some patterns from the data. The next step is to interpret these patterns, the knowledge that we obtain, and finally this interpretation of the results leads to some explanations, predictions, or new knowledge from the data. The most commonly used data mining algorithm are decision trees. These are predictive models that present a good trade-off between understandability and predictive power. So here is an example of a decision tree. There are different types of decision trees. The ones that are predicting numeric target variables can be regression trees, are called regression or model trees. The difference is that regression trees contain numeric values in the leaves. These are the leaves of the tree which contain the predictions of the model, and model trees contain piecewise linear models in the leaves. So the 9 scenarios that I mentioned before are presented here. Here we grouped different chemical and physical parameters in some logical sets. So the first 3 scenarios are comprised only of subsets of the chemical properties of the soil. The 4th scenario comprises of physical properties of the soil. Scenario 5, 6, and 7 combines the sets of chemical properties described chosen in scenarios 1 to 3 and physical properties. Scenario 8 contains all chemical soil properties, and scenario 9 contains all chemical and physical properties of the soil. So for the first part of the study, in the first part of our study, we used only chemical and physical properties of the soil. As I mentioned before, we generated model and regression trees, which were tested by tenfold cross-validation. And here in this table, the correlation coefficient of determination and the root mean squared error are presented. So here we can see that the best results were obtained using scenario 1, where a subset of chemical properties was chosen. And also, almost identical results were obtained with scenario 5, where the same chemical properties as scenario 1 were chosen, but here we also used physical properties. The difference in the model and regression trees between these two scenarios was that in the fifth scenario, the physical properties appeared very low in the decision trees, which means that they have smaller informative power for predicting of mineralized nitrogen in the soil. And here we can conclude that the chemical properties that were chosen in the first scenario are actually more informative for predicting the mineralized nitrogen. And if you're wondering which chemical properties are these, I will show them later. So in the second part of our study, we used only the diagnostic properties. Again, we've generated model and regression trees and tested the predictive performance of the models with tenfold cross-validation. And here the results are presented, and here we can see that we've obtained pretty low correlation coefficient and coefficient of determination, which actually proves that the diagnostic properties are not that informative for predicting the mineralized N in the soil. In the last part of our study, we used all possible soil properties that we had, so chemical, physical, and diagnostic properties. The results are presented in the table here, and I also put the first table where we used only chemical and physical properties for comparison. So here we can again see that the best results were obtained for the first scenario and the fifth scenario. The interesting thing that happened here was that the diagnostic properties did not appear anywhere in the trees. So again, only the chemical properties appeared in the trees, which again proves that these are the most informative for predicting the mineralized N in the soil. But what an interesting thing that happened here was in the third scenario, where in the first case we've obtained very low predictive performance, very low correlation coefficient, which means that this set of chemical properties do not carry any information value for predicting the mineralized N. But when we added the diagnostic properties, the correlation improved, which means that if we don't have any informative if we don't have any data that we can use for prediction of mineralized N, the diagnostic properties can help. But in the other case, where we used only physical properties and we've obtained relatively good correlation coefficient, adding the diagnostic properties to the data set actually decreased the results, worsened the results. And this is an example of the best regression tree obtained for scenario 1, so for the set of chemical properties. And here we can see that only 4 chemical properties were chosen in the tree, which are total nitrogen, organic carbon, and 3 chemical properties were chosen. So this is total nitrogen, organic carbon, and base saturation. This model actually has a very high predictive performance, which makes it very reliable for prediction of mineralized N in the soil. And from this we can conclude that using this type of modeling can we can use only a small number of chemical properties, and we can avoid expensive and extensive soil analysis to predict the mineralizable N in the soil. So to sum up, we've obtained very accurate and reliable predictions of mineralized N using only 4 chemical properties. This reduces the need for expensive and extensive soil analysis, and we've already we have also seen that diagnostic properties help if no chemical and physical properties are available. So the next steps for this study is to use additional data, which includes biological properties of the soil, and repeating these experiments using also biological properties of the soil. And also we would like to try to generate predictive models for larger regions, for example, for EU level. And finally, I would like to thank my colleagues from the Josef Stefan Institute, from the Wageningen University and Chagask, and also I would like to acknowledge the Horizon 2020 project landmark, as part of which this study was conducted. Thank you very much.
Very perfect timing and very interesting talk. I've been a while without using regression trees, so it was very nice to see them again. So do we have any questions for Aneta? No? If not, I have actually 2. One of them is, you were saying in the conclusion that you are going to do that European level.
Yeah.
Mapping. And I was wondering whether or not that will change drastically between different soil types, different pH, whether or not you have checked the role of other chemical properties other than soil carbon.
Actually, yeah, all the chemical properties were used in the in this analysis, but data mining methodology is based on data, so we don't have an influence on what variables are chosen in the models. So these models were actually generated in an automatic way.
And the second question is whether or not nitrogen mineralization is always a good thing or a bad thing, because if you get nitrogen, high pulses of nitrogen mineralization when the plants are not growing, you might lose actually the nitrogen and leach it to the water sources. What do you think about
I'm afraid I cannot answer to this question. I'm a computer scientist, so
all right.
This should be addressed to my colleagues. Yeah.
All right, thanks very much.
Okay, thank you.
Diversity and also stay with nitrogen. So Christian Altersberg is going to talk about habitat diversity strengthens the primary productivity and nitrogen cycling in shallow coastal ecosystems. So thanks, Christian.
I will start right away, or wait a few minutes maybe.
Oh, actually yes, you are totally right.
Yeah.
You're a better chair than
No, I'm feeling the pressure here. Like, yeah. Yeah, I was looking for my co-authors. None of them are here, so that's great. Thanks for the support.
Very nice.
Shall I? Oh, there they are. Okay, great.
So now we can start.
Yeah, now we can start.
Okay, Christian.
All right, thank you very much. So, the title of my talk is Habitat Diversity Strengthens Primary Productivity and Nutrient Cycling in Shallow Coastal Ecosystems. So, yeah, when I uploaded this presentation, the guy at the upload station said, yeah, but with habitat diversity, you mean species diversity, right? No, habitat diversity. Yeah, but species diversity. No, it's habitat diversity. Obviously, this was confusing, so I will see if I can convince you that this is also important, at least to take in consideration. So the system that we've been doing these studies is shallow coastal ecosystems. So they are basically at the interface between land and sea, and they are hotspots for primary productivity, but also a lot of nutrient cycling. And basically, they can consist of a mosaic of different types of habitats. Some of them might like only be like a sandy beach. Sometimes these sandy beaches can also have a three-dimensional structure of huge cyanobacterial mats. They can be like mud holes or have mud in the middle of the bay. They can also be vegetation going on. This is Rupia maritima, so it's a grass growing in the shallow portion of the bay. And if you go a bit deeper, there can also be eelgrass meadows. So sometimes you actually end up with bays having all of these different types of habitats. So usually when you want to try to understand the functionality of these ecosystems, you have to take into consideration that you have actually different habitats working in this ecosystem. So this is an aerial view of one of the bays that we've been working in, and it might be difficult for you to see, so I'm going to help you a bit. But it's actually around 4 habitats here. So we have a very silty kind of mud going on. We had eelgrass meadows, rupia meadows, and also sand. So what we do normally to try to understand this, we go out and we basically take sediment cores and we incubate them either on site or in the lab and measure functions that are to our interest, primary productivity, denitrification, and so on. However, not however yet. So what you end up is you get results which shows both within habitat variation, of course, but also between habitat variation. And this is also a variation that varies with seasons. And then you construct maybe a budget of some sort, but you have to separate that budget on your different habitats. And then you can take that data and try to make a budget for the entire bay. But what you don't take into consideration here is that these habitats most likely are interacting with each other in terms of energy fluxes, nutrients going between the different habitats, especially around the edges. And that might actually result that you can underestimate the functions that you are measuring on the scale of a bay, for example. So this was something that we were very interested in, and we wanted to actually test if this is actually true. Can these habitats interact? So what we know about habitat diversity is that we know that it might actually increase species biodiversity, like butterflies, plants. And of course, we also know that if we have a more species-rich environment, we would expect that to be more productive. Not always, but most of the times. So our the framework kind of was that we wanted to investigate if we change the diversity of habitats, would that also change the functionality in these systems? And this is primarily what I'm going to be talking about at this talk, but obviously we know that functions are not driven by habitats, it's driven by the species in the habitats. And we have also, of course, measured this. But this is, I'll leave this for my colleague Fabian, so he's going to be talking more about this tomorrow. He's right there, right? Yeah. So, but for now, I'm just going to try to establish the link between changes in habitat diversity and how that might affect ecosystem functioning in these ecosystems. So how we did this was that we went out to a bay and collected actual habitats, the 4 most common habitats found in this bay. So we could collect like entire actual habitats, and we transported them back to a field station and place them in this flow-through system. So we could actually create this gradient of 4 levels of habitat diversity. And this allowed us to actually measure the functions in these artificial ecosystems, so to say, in this. I don't want to use the word bucket, but that's actually what it is. So let's call it an artificial ecosystem. Yeah, obviously, seasonality, it's kind of like boring to talk about seasonal effects, but it's still important in terms of understanding the system, right? And this is actually data from the field station during the year that we did this experiment. So you can see how light and temperature varies. So we performed the same experiment 3 times, spring, summer, and autumn. Right, so this is a a picture from the bay, actually, and this is just to illustrate that they can be very productive. So if we look at go to the results here, and we look at net primary production, for example, we can see that there are not a lot of differences between the habitats, but when you start to mix them, you actually get a very clear signal, can be, especially during summer, that you actually get an increased productivity in a more diverse system. And the same was true for the respiration in these ecosystems, and also this 24-hour net oxygen flux. So that is the sum of consumption and production of oxygen. Basically, it indicates if the system is turning more or less heterotrophic or autotrophic. So in this case, the system turned more autotrophic when you had an increased habitat diversity. We also looked at several nitrogen-related processes, not going into the methodological issues here, but we could see for the fluxes of ammonium that there was a tendency for the system to keep the ammonium when you had a higher diversity. Nitrogen fixation was extremely positively related to the diversity of habitats, whereas the nitrate plus nitrate flux, I mean, the concentrations of the nitrate are fairly low in these systems always. And denitrification, it's more of a sensitive process. It's divided in several steps. Did not respond to changes in habitat diversity. And as I mentioned, primary production was indeed significantly positively affected by diversity of habitats. Also then, of course, the autotrophic biomass, in this case illustrated as sediment chlorophyll A. So to take this one step further, what we did was that we calculated the net diversity effect, basically comparing the expected diversity effect based on the monocultures, monohabitats in this case, compared to the observed effect, comparing mono with the highest diversity. What we can see here is that we have this net habitat diversity effect both for the biomass, the productivity, the respiration, and also the 24-hour net oxygen flux as well as nitrogen fixation. So this indicates that there are some kind of interactions between the habitats that results in this positive effects. I mean, this is purely like, it's only, that is really bad graphics. Okay, so I mean, it's only speculations, but obviously we kept the temperature and light environment was the same. We can't capture all of things happening in these systems, but I think it would be fair to say that there were some kind of nutrient exchange going on between the different habitats that actually resulted in an increased productivity in these systems. So we wanted to take this one step further, mainly to illustrate the importance of habitat diversity on the base scale. So lucky for us, we of course took the GPS position of all the cores, and we also had habitat maps available as GIS layers. So we spatially modeled all of our ecosystem functions around each of those positions where we actually measured. So if with a buffer zone of 50 meters, so if one core interacted with another core within this buffer zone, would we have an effect of habitat diversity or not? So this is the model output from that analysis. And what we can see here is that the observed number of habitats surrounding each of those different sampling points was positively related to the 24-hour net oxygen flux and the net primary production, basically illustrating that we could expect to have a higher productivity on the base scale. So the A and B, that will illustrate the productivity in this bay, taking the habitat diversity into consideration, whereas C and D are the null model, and E and F is the difference between the two. So adding the number of habitats to a model with only habitat identity increased the explained model then from 20 to 30% roughly. Okay, so to summarize, habitats can be functionally different in terms of primary productivity and nutrient cycling. Coastal ecosystems with a diversity of habitats influences productivity and cycling beyond what an ecosystem of a single habitat would contribute with. So this, we interpret this habitat diversity effect that would be equivalent to the over-yielding in a diverse species assemblage. So some kind of evidence for positive interactions among these habitats, which could be good to at least think about when you try to estimate functions on larger spatial scales. Yeah, it's
Really great talk. Do we have any question? Yeah, for some reason I knew you would be asking that one.
So coming back to what you said at the beginning, you obviously manipulated habitat diversity, but I guess you also manipulated species diversity.
No, we did not.
But I mean, you must have, presumably you had higher species richness in the treatments with more habitats.
So,
are you sure that it's habitat diversity and not species diversity?
so this is gamma diversity. And as you can see, there are a slight increase of the diversity in our system when during summer, for example, where we observed the largest effects. What we did was that we actually modeled taking both the gamma diversity and the habitat diversity into considering a structure creation model framework, so we could compare the relative contribution to both of these. And habitat diversity was the strongest, was predicted to be the strongest driver for the functionality.
Is there any kind of like hormonal signaling going on between any of these organisms?
Hormonal?
Just something in some
Oh my God, I don't know.
Because they're all obviously in the same water,
Yeah.
aren't they?
Yeah.
Because otherwise, it's really neat, but I'm just struggling to see what the mechanistic explanation is.
No, I mean, I think it would be fair to say if you have a system where, because the biochemistry and the physical and the chemical, as well as the biological components in these habitats, they differ a lot. So, I think it's fair to say that nutrients can move up and down the water mass, and it can be exchanged with other habitats that actually might need that nutrients more than others, for example. So it can be purely driven by diffusion, for example.
This water's recirculating.
Yeah, so the water was moving like constantly, of course. Yeah. Yeah.
Any more questions? Yeah.
Did you try to use some other measures of habitat diversity, not just number of habitats?
Yes, we did.
What did you use?
Did
you use any which respected the abundance between the surface of each of the habitat?
So what we did was that we measured, of course, a lot of stuff in these habitats, like organic material, everything that you can measure, basically. And we, okay. So what we did was that we plotted this is a PSO plot, right? And we took those all those different values and compared them to each other and transformed that. So we had, instead of having habitat diversity as a categorical variable, we turned it into a continuous variable, because then we can also take into consideration the within habitat variation, so to say. But we end up with the same results, basically. Yeah.
All right. I have a question regarding to what Eric was suggesting. Like, I know you tested the relative importance of habitat diversity or local species richness, but do you know if they interact? Like, whether or not you have a higher diversity of habitats which at the time are also rich in a species, you know if there is an interaction on the provision of
if the habitats are also interacting in terms of species, you mean? Like
yeah, if you have like high diversity of habitats and at the same point these habitats are high in species richness rather than low, you know if there is an interaction between these two factors?
No, I don't know that, no. But that would be a fair assumption, I guess, that there could be something going on as well. Yeah.
Okay, thanks. Well, thanks, Christian.
Thank you.
Next speaker is Ali Birket. All right. Christian is a fairly big guy. So Ali is going to talk was about litter manipulation and how it alters the stoichiometry of soil enzymatic activities in a lowland tropical forest. So thanks, Ali.
Thanks, yeah, lots of long words, I realise, in my title. So I'm a research associate at Lancaster University, working on a large project looking at litter effects on soil processes. And what I'm going to present today is just one part of the work we've been doing. It's very hot off the press kind of data. So I've actually put on the slides with the graphs on, I've actually put the please don't tweet symbol. Just please don't tweet the image. You can tweet what I say is fine, but don't tweet the image just because we've obviously just produced this out of R, so it'd be great if we could keep that quiet for now. So one of the main reasons that we're looking at litter effects on soil processes is because there are predicted changes in litter fall patterns in the future. So there are predicted phenological shifts with altered rainfall distribution, with temperature, and also with other sorts of climate effects like storms, as a colleague was discussing in his talk earlier. There is increased growth in plant productivity is predicted with atmospheric CO2 rising, and that will obviously have an impact on litter fall. However, the potential impact of these changes in litter fall on below-ground dynamics has been largely ignored in comparison to lots of the other studies that are out there looking at other effects. So what we've wanted to look at here is, will altered litter inputs affect soil carbon and nutrient cycling? The reason for that is because litter fall clearly has a strong effect on below-ground processes and is a link between the above-ground and the below-ground systems. The litter fall is adding substrate into the soil system, and the soil microbes are producing enzymes to acquire carbon and nutrients from that organic matter, so they're breaking down that litter. The carbon that they're using is also being used for enzyme production, and those enzymes are then being invested in further nutrient acquisition. So it's expected that production of carbon-degrading enzymes will be related to the amount of substrate available. So where there is more litter, there will be more activity of carbon-degrading enzymes breaking that down. But the nutrient-acquiring enzymes are likely to be produced in response to requirement. So when a microbe is feeling short of nutrients, then they will produce more enzymes to acquire those nutrients. Because of the involvement of carbon investment within this process, there is a strong relationship between carbon and nutrient acquisition within the soil. That strong relationship was shown in a 2009 paper on a global scale, looking at a variety of different ecosystems across the world. On the left, you're looking at nitrogen acquisition in relation to carbon acquisition, so carbon is on the y-axis. And on the right, that's phosphorus acquisition in relation to carbon acquisition. So you can see that strong relationship on a global scale. In understanding that, I was thinking of an analogy. I like my analogies to get my head around these processes. And my favourite board game is Settlers of Catan. And I realised that basically what we're talking about here is investing resources that you're requiring in getting further nutrients. So in the game, basically, you're trying to expand your empire, and you get some resources, but you need to invest those resources in making settlements and making developments. So if your initial resources are your carbon, you're taking your carbon and then investing that in getting your nitrogen and your phosphorus and all of your other nutrients further on in the game. We wanted to look at whether this relationship has been affected or will be affected by changing litter inputs. And in doing this, we used samples from a tropical forest here in Panama. And it was a long-term litter manipulation project that we were using. So there are, across this forest in Panama, there are 15 experimental treatment plots. And they're trenched around the edges of each plot so that you're not getting sort of flow of substrates and things from one treatment plot to another. It does come out clear. So our treatments were applied within these plots, and there were 3 treatments used. One was the standard control, which is shown in the blue, which is just litter left as it naturally falls. And then we have a litter removal treatment and a litter addition treatment. The litter removal treatment involves raking up the litter from one plot and then adding it to the litter addition treatment, which does feel as daft in the field as it sounds. But that creates our three treatments, and you're using the natural litter that's found within the system. What we were hoping to explore using the data from this large experiment was these set of hypotheses. So we were investigating the relationship between the litter treatments and the carbon and nutrient acquisition. We were specifically wanting to look at whether the activity of the carbon-degrading enzymes will be higher in the litter addition plots, as you would expect if the carbon-degrading enzymes are increased with more substrate. Whether the nutrient-acquiring enzymes would be higher in the litter removal plots, so this is where there is less substrate, again tying back into the literature, and overall whether the litter manipulation treatment would alter the relationship between the carbon and the nutrient acquisition. In doing this, we used soil samples taken after 5 years of the treatments being in place, and we measured the activity of 4 different enzymes, 2 of which involved in carbon acquisition, 1 in nitrogen acquisition, and 1 in phosphorus acquisition. So this is where we come to the data. And initially, we first of all just wanted to look at whether the activity of the enzymes overall varied by litter treatment. And as you can see on my I still can't say it the right way around, NMDS plot, where the yellow plots are the litter addition treatment, the blue and purple are the litter removal and the control plots, and you can see that there is more similarity between the litter removal and the control than there is the litter addition. So this looks like there is an effect of treatment in that litter addition seems to be performing differently to the others. The difference on the first axis seems to primarily be explained by the phosphorus activity. To look into how this is affected by the soil nutrients in the soil system, we use specifically the data from the growing season, because this was when we had soil nutrient data available. And you can see again, even just within a subsection of our data, you're still getting the same effect of the litter treatment. But interestingly, although the soil phosphorus is having an effect on the data overall, it's not actually explaining that split along the x-axis. It's not explaining that difference in those two treatments. This is suggesting that with all of the different nutrients we looked at, they're not having as much of an effect on the enzyme activity as the addition of substrate. To look into the specific enzymes, this plot shows the different sampling, the sample statement at different times against 2 different carbon acquiring enzymes. So you're basically looking at carbon acquisition at the different time points. And at the end of the experiment, I'm going to see if I can use this. Yes. So at the end of the experiment, looking at after 5 years, you can see that in both carbon acquiring enzymes, you have the lower carbon degradation enzyme activity in the litter removal treatments. And in the earlier samples, so there may be some temporal effect, but in the earlier samples, when you're looking at the nutrient acquisition, you're getting the opposite effect. You're getting the lower nutrient acquiring activity in the litter addition treatment. But the really interesting result is when you look at the relationship between carbon acquisition and nutrient acquisition. So on the left, you've got phosphorus acquisition across the x-axis and carbon acquisition up the y, and on the right, it's nitrogen against carbon. So where in the control treatment, you have a significant relationship, so the blue lines in the control treatment, is a significant relationship between enzyme activities. That is also a significant relationship within the litter removal treatment. However, in litter addition, that's no longer a significant relationship. There isn't a strong relationship anymore. So what we're suggesting here is that the amount of litter, the amount of substrate available, is having an impact on that relationship between carbon acquisition and nutrient acquisition. It's a weakened relationship when litter is removed, and a stronger, no, I've done that the wrong way around, haven't I? It's a stronger relationship when the litter is removed and a weaker relationship when litter is added. There we go. So to summarise, now I've just fluffed that, to summarise, we've seen that there does seem to be some evidence that there is a greater activity of carbon-degrading enzymes when litter substrate is removed, and there is some evidence of a decrease in nutrient-acquiring enzyme activity when there is litter added, when substrate is increased. Those results, I think, need looking into further because there did seem to be some sort of temporal effect as well. But that's a sort of a working result. But what seemed to be most clear was that the litter manipulation did alter the carbon cost of nutrient acquisition. It altered that relationship between carbon and nutrient acquisition. And just my acknowledgement slide. Excellent, I'll run away.
Yes?
So it was done as hang on. The bottom, the middle one of those references is the one with the protocol that we use. So it's a soil slurry, basically, that's made, and then you're then adding a substrate analytically to that, and then measuring the activity within a lab setting. So we weren't actually extracting it, we were using the soil slurry. No, so I'm working as part of the Forest Prime project. My boss is standing at the back. So I'm working as part of MSA's Forest Prime project, and this work is following on from her previous research in Panama, but it might be an interesting group for us to have a conversation with.
I have also a question. I thought it was kind of interesting that you have like 3 years of data, but that was after 5 years of experiment, or more or less in the middle. And you only find this drastic response at the very end, it appears. And I was wondering if you think it's a threshold behavior of the soil that litter addition is enriching the soil until it arrives at a point that the soil microbes react differently to the nutrients, or do you have any hypothesis of why you have right at the end?
I haven't formulated a hypothesis. My co-author is waving at me from the back, so she might be able to answer for me.
Yeah, I was wondering if it would be about that, because the soil microbes should react very rapidly to the addition of extra food.
That's what we thought, maybe.
All right.
We've done a similar experiment in a slightly different site, but the same principle, and we're not finding any effect after 3 years of treatment. We're not finding any effect at the moment. So.
Yeah, thanks. Okay, Malik. So, last talk, we still have 1 minute, so don't get started. Sabine Reich, if I pronounce that properly. She's going to talk about responses of ecosystem carbon dynamics to climate change, recent advances.
I try to. Don't worry. Won't really happen. Well, if
you guys can grab a seat, there is plenty of space. It's all right? Okay. Yeah, Farino, what's yours?
Yeah, hello and good afternoon. Thank you for staying and not just running away. Welcome to the last talk of the session, and for the ones that have been here in the beginning of the session, this talk will link to the first talk again on more focused on carbon, or entirely focused on carbon. So we'll be talking about the responses of ecosystem carbon dynamics to climate change, and especially the recent advances. as we all know, climate change is happening. I don't have to convince anyone here. But the recent IPCC report shows that we will have increased surface temperatures, and we will also have changes in average precipitation. But we are not only interested in, or that it happens, but we also need to know what the feedbacks or the ecosystem feedbacks to these changes in environmental conditions are. In order to do this, we need long-term climate change experiments to help these models to do better predictions of what we predict for the future. And we have one, or in CEH, we're running one of the long-term experiments in Wales, which is a climate change manipulation experiment where we exclude rainwater to simulate drought during the plant growing season for a relatively long period of time. We increase the air temperature in these systems by passive nighttime warming. So we have curtains covering the plots, so we reduce the heat loss, so it's passive nighttime warming. And then we also have control plots to see what the change is relative to ambient conditions. Yeah, we're in Wales, so it's an upland heathland pot soil, and it's actually a very interesting system because it's not just an organic layer, but it's an organic layer on top of a mineral soil. So we have, for quite some time of the year, that the soil is completely filled with water, which will be important later in the talk. And, well, at the beginning of the experiment, people would think, why would you try to put a drought on such an ecosystem, because it's wet anyway, and we have a lot of rain in Wales, some right now. However, this is only one experiment in a climate change network, and the reason, or the name at the moment is INCREASE, and it's a suite of 8 shrubland ecosystems across Europe, where we have 2 gradients. One gradient is from the east to West Europe, which is a precipitation gradient, and you see the UK is further up on the very wet side. And we have a north to south gradient in temperature. But then we don't only have the European scale, but we also have these kind of experiment on a global scale. And I will show results from country scale to continental and global scales. And as I said before, I will be focusing on the carbon cycle. So we have above ground biomass, we have below ground processes, which you can study in detail. And then, well, as an integrated measure, often we measure soil respiration to somehow integrate what's happening below ground, especially in these long-term climate change experiments. We cannot just dig holes and then see what's going on if we want to have these experiments going for decades or even longer. So the questions I will be focusing on is, first, do above-ground carbon pools behave like below-ground carbon pools? Second, how much soil carbon is lost due to climate change? And lastly, what drives carbon loss in UK, especially the wet soils? The first study is on above-ground biomass. Mark Estiard collected data from I think, European shrublands, where he focused on the drought treatment. So on the x-axis, we have the annual precipitation, and on the y-axis, we have the above-ground biomass productivity, and this is a flux, so it's a yearly measure. Then on the left-hand panel, you see all the data points, and then if you go to the right panel, we see for each side, you have this gradient from where he put both control and dried, the results from both control and dry plots together. And you see that they actually fall on one line. So we have a decrease in above-ground biomass productivity with drought, and this is basically predictable from a precipitation gradient. However, when you look, the yellow arrow shows where our side would be. So we have the heathland side, which is low productivity in an actually very wet ecosystem, so this falls somehow off this scale, and I will go further into it later. This year there was another study conducted by Andreas et al., where we looked into the above-ground production across biomes, which is highly dynamic in response to drought and warming. And we also see different responses in the short and the long term. And this is just a reminder that we need to run these studies for a long time. So on the x-axis, there's the treatment year from 0 to 18. And again, the yellow arrows show where our site, where the data contributed to. And on the y-axis, we have the treatment effect, which is in comparison to the control. And we see for the drought and the warming, they both behave differently when it comes to above-ground biomass productivity in response to drought and warming. But then we also have a switch, so ecosystems, the above-ground biomass seems to adjust after a certain while. So if you only run an experiment for a certain while, then, well, you might misinterpret or you miss half of the story. Moving on from above-ground biomass, what happens in this integrated soil space, what we often measure is soil respiration. Joanna collected data and looked into the response of soil respiration to warming on a global scale. And surprisingly, the temperature response of soil respiration to warming was largely unaltered across biomes. So on the x-axis, we have the temperature, so it's a temperature range from 0 to 40 degrees to capture all biomes. And on the y-axis, we have the logarithms of the soil respiration. In blue, we have the respiration values from controlled, and in red from the warmed plots. And what we actually expected was that we see patterns, so clouds of red and clouds of blue points, and we didn't think they would actually overlap so closely. However, there was one only the desert ecosystems behaved differently. You can read this up in the publications in PNAS. However, our data then contributed to the northern shrubland sites, also back into this cloud, which actually, when you see the temperature range, is relatively narrow compared to most of the other biomes. So having the information of the above-ground biomass and the integrated soil flux, what happens to the actual soil carbon stock? And our most recent publication was on soil carbon stocks as affected by warming. And this just was published in Nature beginning of the month, where Tom Crofter put an analysis together on the standing soil carbon stocks, and we could say that under warming, the soil carbon stocks is likely to decrease. And we made this statement that 55 trillion kilograms of carbon is lost from the topsoil by mid-century, which is quite an essential amount of carbon lost from the soil. However, when you look into, so just an explanation, when you look at this graph, we have the standing soil carbon stock on the x-axis. So the higher the carbon stock, the more carbon you can lose. And our side is one of the driving points because we have a peatland, so there's a lot of carbon, and where you can stimulate activity. And on the y-axis, we have the change in carbon stock measured as kilogram per square meter per degrees of warming per year. So putting all the data together, we can conclude that we actually have a decoupling of above ground and below ground carbon pools, because the above ground, we said we can predict from the treatments, and well, the soil respiration didn't show a change, and then we actually see that we lose carbon. So it is important to not just do this disintegrated meta-analysis, but actually put the data together where you have one ecosystem, where you measure above and below ground together. And this is what we did in these European sites, where we looked, okay, what happens above ground and below ground at the same time. So looking at the x-axis, it's an aridity gradient across Europe, taking the precipitation and the temperature into account. So on the left-hand side, we have dry, warm climates like Italy and Spain, so the southern sides. And on the right end, we have the wet, cold sides like the UK. Up there, our point's doing something different. And on the y-axis, again, we have the change compared to the control, so to ambient conditions. We have two sets of points, and here we have the grey point, which is the above-ground biomass productivity, and they're in grey because they don't do very much over the time well, they vary, but we cannot say they do one or the other. And in terms of soil respiration under drought, we see that soil respiration is driven by soil moisture, which actually show a response when you go along this aridity or this index of climate across Europe. However, this weird peatland soil that's partly waterlogged in the year does something different. And we looked into it, what might be driving this end of the curve. And we concluded that it is due to a change in soil structure. So this is something called the soil water release curve for people that are not familiar with it, as I am, so I try my best. On the x-axis, we have the soil water potential, and on the y-axis, we have the volumetric water content. In blue are the measurements for the control plots, and in red, the measurements from the drought plots. So you have a core that dries. If you dry it, and you see how fast the water goes through, or actually it's a measure of how good the soil can hold the water. And we see for the drought plots, it can actually hold significantly less water than the control plot. So, and this is because we have a boost of CO2 from the soil because of drying of the soil. So the soil moisture retention decreased by 10% with drought, and then we got an alternative stable state of soil moisture in our system. So to sum this up for you and to give you the take-home messages for the first question, actually, do above-ground carbon pools behave like below-ground carbon pools? Actually, not in the short term. We rather think they are decoupled. Second, how much soil carbon is lost due to climate change? Well, a significant amount, and this is linked to how much carbon you actually start up with. And the last point, what drives carbon loss in UK wet soils? We think it's a fundamental change in soil physical properties, so we shouldn't forget about it. Yeah, and then thank you for your attention. Thanks to all the people maintaining the sites, funding these long-term climate change sites, and to the lead authors of all these meta-analysis I just presented.
Thanks very much. Do we have any questions? Yep.
Sabina, thanks. That was really, really interesting. Have you got any explanations for why the actual soils become more hydrophobic with drought? Is it that it's something to do with organic colloids?
Actually, we think, actually we think it has to do with the microbial community and fungi, that when the soils dry out, they can make hydrophobic surfaces, so they can just push the water through. So this is the next step where we're going. We will look into the microbial community and how, well, we know it has changed, but we need to do a reanalysis of our data to see what's actually happening. So we think it's probably the fungi that make the soil more hydrophobic.
So they're becoming ecosystem engineers?
Actually, yes. Yes.
Taking it into a slightly different area, would you therefore expect this to change catchment hydrology with potential impacts on runoff and potentially flood events?
I think so, yes. That's probably the easy answer, right? Well, these are natural, from the side, that's a semi-natural habitat, but this is what you create when you put ditches in the peatlands anyway, so you create a preferential flow, so you will get different hydrological properties, yeah.
So you were actually having the macropores opening up, or,
so part of the soil physical study of this part is that we have this organic mineral soils, we have this organic layer, and then we have the mineral soil. And part of this story is that this mineral soil was really thick, and then there was a heavy drought event in 2005, which made the soil crack. So we must have had very thick cracks. So instead of getting waterlogged, or the soil stopped getting waterlogged, so we had probably not not a run-through of water, but we can see that the soils just don't wet up, and this is because of cracks. So yes, you have preferential flow created there. So that's why the organic mineral soils are really important, because if you have a drought, then you can fundamentally change the soil physical structure that changes your whole hydrology, which changes, well, has impact on plants and microbes and everything.
It opens up all sorts of possibilities. Yeah.
Yes, which is quite interesting, is just to figure out what's actually happening.
Okay, thanks very much. Thanks to all of our speakers and for you to attending.
Hello.
I never know how to say like yeah. No, it's very yeah, you keep saying I'm very well though. Right, okay, follow your blog. I think you are quite thinking of that. I need your sensitivity, you are not gonna, you know, no, that's the yeah, I always get confused when yeah, what's up? Yes, yeah, that's the reason I get back yeah, so I do. I do the same, I do. Just a lot like the other.
Yeah, because the Germans always honest, but the Swiss kids do.
Yeah, but they're not doesn't like it. I didn't want to doesn't like me. Not like me. Oh, yeah, sorry. Yeah.
And that's probably based on my knowledge of our experiment. But thank you for the question. Yeah, it's something we need to be included.
Oh, yeah, I mean, I've accidentally. All right.
Yeah, but you're doing well.
Yes.